All right, boys and girls, let's get started for today. My glasses back on. We want to give a little brief review. And remember, this does not replace watching the full lectures and taking notes, but a little bit of talk about I believe it's chapter 17, right? I think your book calls it From Gene to Protein, which is a big topic, quite frankly, right? So remember, the genes are the code of the bases, right? Basically the sequence of the bases on or in, whatever you want to say, in the DNA, right? That's what makes up the genes. So I'm going to try for big picture today to help you, because I understand you can get very bogged down in the tiny details. And when you get to the end of one of these sections, then you're kind of just like, wait a minute, what was this about? Right? So I'm going to try to give you, again, a big overview. So when chapter 17 says genes to protein, this is the connection from the DNA to how we actually make all the proteins that do all the things in the body, right? To get to the protein, we have to go through, the DNA is not directly turned into protein. We call it the central dogma. Well, I never called it that, but people did, right? So DNA to RNA, oh, look at that, I made a little arrow, to protein. Now, this is, I can't spell protein today, it's EIN. This is not always the case, as we know from viruses in particular, and as we know from evolutionary biology. We're pretty sure that the world was an RNA world first. So I'm going to circle that in red, that originally cells went from RNA directly to protein, to functioning things. And DNA was sort of a later invention evolutionarily, if you want to think about it that way. It was a way for the cells to store their hereditary information. Because the RNA is getting used so frequently, it's much more susceptible to damage, right? So you can think about DNA as really a way to store the genetic material and a way for it to be kept inside the nucleus if it's a eukaryotic cell, so that it's less likely to get mutated. Obviously, it's going to get mutated, but that's less likely. So if we go back to this story here, where I was telling you about DNA to RNA to protein, right? And let's remind ourselves all the things that proteins do in cells, okay? So I'm going to put a text box down here. And now we're kind of in a better place to go back and actually think about all the things that we talked about that proteins do in chapter 5. Okay, so if we go back, and remember, I told you chapter 5 is never going to go away as much as you don't like it. So obviously, proteins are enzymes. So think about all of the metabolism that we discussed in chapters 8, 9, and 10. Remember, all of those enzymes are proteins, and they all have to be manufactured from RNA, from code that you have in your cells in the DNA. So all of metabolism, all of the proteins that are involved in not only metabolism enzymes, but I'm going to put on there let's say transport, right? All the things to get us through the membrane. So all of chapter 7, moving molecules from inside the cell to outside the cell, and from outside the cell to inside the cell. All of the structure that we always say, oh, this is a protein, right? So cell membrane, phospholipids plus proteins, right? Bone is constructed partially of proteins. Muscle, obviously, constructed of proteins. So any type of motility made of proteins. All of the cytoskeleton, think of chapter 7 again, or chapter 6, tour of the cell, right? Think about spindle fibers, because that's most recently on our minds, right? All of the cilia, all of the flagella proteins, right? All the spindle fibers, all of the connections between the nuclear lamina, between the outer and inner membrane of the nucleus, storing things, storage of amino acids, right, to use for later, transport, structure, all of our defense. So all this talk about immunoglobulins, which people slang as antibodies, right? Which we're all talking about now that we're talking about COVID and getting vaccinated. We're producing immunoglobulins and antibodies and other what we call cytokines. They're all proteins. Most of your hormones, remember, are peptide. Most of them are peptide hormones, which, remember, peptides are short proteins. Most are peptide proteins. And again, this isn't something I'm going to ask you on the exam per se, but this is something to help you remind you how important proteins are. Remember, sort of really flippantly or facetiously in chapter 5, when we got to proteins, and I said, okay, what are the functions of proteins? Proteins do everything, right? And I kind of chuckled. And I know that wasn't helpful when you were in chapter 5 trying to memorize all the functions of all these different organic molecules, but it's true. In essence, proteins do everything, and proteins control how your cells make all those other organic molecules, right? Think about metabolism in the big sense, not just breaking things down, but also building things up. How do you build muscle? By adding amino acids to make a chain of proteins. So this is what we're focusing on here in chapter 17, and it is crucial for life, right? For a cell, for one of the functions of a cell is to be able to convert the stored code of DNA through RNA into protein. Now, we are only going to focus on predominantly the messenger RNA. So if we're looking at this DNA to RNA to protein, we are only really focusing on messenger RNA predominantly, but in your handout for this chapter, you'll see that I listed out at least 2 other types of RNA. And there are about 17 or 18 different types of RNA, depending on the kind of cell you're in. And many of those RNAs are actually more than just code. Many of them are functioning enzymes as well, and many of them regulate a lot of the protein production that we're actually talking about. So it just gets more and more complicated. Remember when I told you when we were reading chapter 7 and we were talking about the different functions in the cell, I said there's a whole book that's as big or bigger than your textbook that's just called The Molecular Biology of the Cell. And each of the parts, each of the sections of chapter 6 would be many chapters, as well as many volumes of scientific research on all of these different types of RNA. So we're going to focus on just messenger RNA, ribosomal RNA, and transfer RNA. And these are like the 3 baby. These are just the barely kinds of RNA that you need to know about, right? And I'm not going to go through all of the characteristics of them because they're listed in your book, and they're also listed in the handout that I sent, that I put in this module, right? So the M stands for, just briefly, messenger RNA, right? The R for ribosomal, which is important structurally to the ribosome. And it's important also because ribosomal RNA is pretty much the basis of how those 3 domains or 3 different cell types, right, were established for classification or for taxonomy. This is the basis of those 3 domains of archaea, bacteria, and eukarya. It's actually differences in ribosomal RNA, how cells make proteins, the structure for how those ribosomes make proteins. And then the transfer RNA, this is the go-between. You can think of it as the translator. This is the RNA that has that weird sort of 3-dimensional shape. that basically goes to the ribosome to convert the messenger RNA to peptides, because it carries the amino acids. And I'm just going to abbreviate the amino acids as A.A, right? Carries the amino acids to build that protein chain. So you'll see that the book, this chapter is kind of divided into two parts. The first part is about transcription. Actually, it's three parts. The first part is about transcription. And transcription is a process of transcribing or converting a gene or small group of genes into messenger RNA, right? So that whole first part of the chapter, trying to make that long, so it just takes up, like, there we go. That whole first part of the chapter about the enzyme RNA polymerase, promoter sequence on the DNA where that RNA polymerase can bind, and that what we call the transcription bubble, right, of the process of how only, remember, one strand of the DNA, the template strand, is, I'm going to put this in quotes, is read and converted to transcribed into messenger RNA, and that would be the 3' to 5' strand, because the message is going to be complementary and anti-parallel to the DNA. So the messenger RNA is going to be read from 5' to 3'. It's going to bind to the ribosome on the 5' end and be read 5' to 3' into amino acids. So that 5' to 3' orientation is important because that comes up over and over again. And you'll remember from our discussions last week that the 5' end has that phosphate group on the 5th carbon as well as the 3rd carbon, and a 3' end of nucleic acid, a chain of nucleic acid, has a hydroxyl group on the 3' on the 3rd carbon, not a second phosphate, but it still has a phosphate on the 5th carbon. Right, so that process of transcription is important not to get that confused with the process of translation. The process of translation is using a ribosome. That's like the meeting place, right? Using a ribosome, the structure of a ribosome, that's where our messenger RNA and transfer RNA are going to meet and build a polypeptide. And this process of translation, I have it described in basically 5 steps. Yes, you need to memorize those steps and know what happens in each one of those steps. If you watch the video for this, you'll see me drawing it. You'll see me going over it multiple times. I would suggest you do the same thing, get some blank paper, memorize it, draw it. Why? Because this is going to come back. You're going to be asked this question, describe how a protein is made from DNA into amino acids. You're going to be just asked to tell about it in anatomy and physiology. You're going to be asked to repeat this process in microbiology. We're just going to add some more details. You're asked to describe it in genetics class. So this is a key, replication, transcription, translation, these are key processes, just like chapter 5 was key about those organic molecules and chapter 6 was key about the structures of the cell, the eukaryotic cell. This is going to come back to haunt you again. So if you memorize it now, it's going to help you later, right? So I would practice these 5 steps. I would draw them. I would get a piece of paper and draw them and say them out loud and repeat them. That I can't spell, over and over until you can recite them and describe what happens in translation. One more thing, though, I would mention about transcription in eukaryotic cells. There's a section there in eukaryotes only, we call the messenger RNA gets processed. And the processing, couple of things. We call it a 5' cap is added, a 3' poly A tail, and splicing. Because what happens is there are gaps in the transcript. So the DNA has sort of spacers, or what we call junk DNA, or introns, is their technical name, and those introns need to be cut out or removed. And the exons need to be put together to get your sort of final, and it's a little bit shorter, obviously, messenger RNA transcript that is ready for translation. Now, the actual translation, there's the process of translation. That's what I'm talking about, those 5 steps. And then there is also the mechanics or the logistics, if you will, of the alphabet. How do I spell that? That is that chart that is the in your textbook that shows a messenger RNA codons, right? And remember, a codon is a set of 3 messenger RNA bases. It's actually just a set of 3 bases, I should say. It doesn't really matter if it's DNA, RNA, or any of the types of RNA. That messenger RNA chart, it's the same chart in every textbook ever. You do need to have a copy of it with you when you're taking the quiz on Wednesday, because it you need to use it in order to tell me, for example, if I give you a sequence of messenger RNA, you need to be able to tell me what amino acids it codes for. So you do need to be able to use that chart. And then the last part of this chapter is about mutations, which are any changes to the DNA. And we limit it to the changes in the DNA. In Bio 1, we'll talk about messenger RNA mutations, which aren't as frequent, but there can be mutations during transcription. But in Bio 1, we stick to just any changes to the DNA. And what would cause those changes of mutations? The causes are simply random, right? We call spontaneous mutations. So those are mutations that just happen. The spontaneous mutation rate, meaning how well DNA polymerase 1 repairs, and those SOS enzymes find mistakes in the DNA and then replace them during proofreading. It leaves about 1 mistake in a million bases, which translates to about 1 codon mistake or 1 base mistake every 1,000 genes. It might be in an intron, so it might not make any difference at all. Most mutations are what we call silent, and it doesn't make any difference, but sometimes a mutation is very dramatic if it's in an exon and if it's in a key part of the expression of a protein or the function of a protein, maybe where a protein folds, maybe where an enzyme makes its active site and it's different. It could have a very positive effect. That enzyme could have a new function, or it could have a very deleterious effect. Maybe it inactivates that enzyme. However, causes, that's just spontaneous mutations. And then there are mutagens, which are forces, factors, when I think of them, both physical, like radiation, normally what we think of as a physical mutagen, or a chemical. Those increase the mutation rate. And people measure that and, write research papers about that. And right, those are things that shouldn't be in our food, for example, or the things that we drink, or the water that we consume. And then there are categories of mutations based on the outcome in, let's say, in the protein, right? So the change happens to the DNA, but that can alter the RNA, which of course can then alter the amino acid that is going to be put in the chain of DNA, which can then, of course, alter the function of the protein. And those categories, we call them base substitutions or point mutations. Well, point mutation just means one place, right? Base substitution is pretty self-explanatory. One base is substituted for another. And then the result can be things like a silent mutation, where no change to the amino acid. It can be a missense mutation, right? So a change to the amino acid or a change to the base, but we still code for the same amino acid. A nonsense or a different amino acid mutation that causes a stop codon to be put in. And then there can also be what we refer to as a frame shift mutation. And that can be either a base substitution, frame shift mutation, put that out here. Because that's usually one or more bases inserted or deleted. So that's going to change the reading frame is what that means. So it's going to change all the amino acids that are produced from that particular sequence of DNA. And again, it could turn out to be a beneficial thing to the organism, or it could be a very deleterious thing for the organism. I think we have this connotation, this idea that anything that's a mutagen or a mutant is bad for the organism, but it actually is an opportunity as well for new proteins to be formed or new functions of proteins to be come into being, if you want to think of it that way, or to come into the gene pool so that it can be used by an organism for a new function that that organism couldn't do before. Okay, so I'm going to stop that share, ask if you guys have any questions that come to mind immediately or anything that you're concerned about. And I would suggest watching those videos, the full lecture videos, taking notes, using the note outlines, and practicing to get a full understanding of the chapter. It's a lot. That's again why I'm giving you a couple extra days here. We're not having this quiz until Wednesday, but I'll just open it up right now to questions that you might have. Go back to the gallery. Yeah. So far, yes, no, maybe so.